Hey students, it's Mr. Lucido. Just wanted to create a video for you that debriefs the free response questions from your personal progress check. I went over one of these in class, but I'll also be going over the other one that I didn't get to in class in this video. So hopefully you find this helpful. Reach out if you have any follow-up questions, but again, be patient with yourself as we're getting used to some of these questions from College Board. So we're going to start with the one that I went over in class. So I'm going to use just a little highlighter here like I did in class to highlight just the main things here. So it says scientists are studying the structure and function of receptor X, which is this single polypeptide protein that is found in the membrane around certain types of cells. So it's a single polypeptide chain that's found in the membrane of a cell. Receptor X contains no alpha helices or beta sheets. We should be thinking secondary structure right there. A specific molecule outside the cells is recognized and bound by receptor X. The binding of this molecule to receptor X causes the cells to have a particular response. So there we see what the function is of this protein. To study the structure and function of receptor X, the scientists are altering the sequence of small sections of the receptor. Both the normal sequence of a 5 amino acid section of receptor X and an altered sequence that contains 2 amino acid substitutions are shown in figure 1. So we've got our normal and we have our altered. Okay, so let's see if we can identify the two differences. So remember, the only unique thing in an amino acid is the R group. So that's what we're going to focus on of what's changed. I said there's 5 amino acids in this section. And so let's look for the R groups. So the R groups are, here's one. We can see this amino acid, all right here, has the carboxyl and the amine group. And there's our hydrogen that we're used to seeing, and here is our R group, right? So there's our first one. Now we can just go down the chain. So this is our second R group, our third R group, fourth R group, and the R group of this last one is actually just a hydrogen. It's a very small R group. Okay, so those are our 5. Let's compare it to the altered. And so this R group is the same. The second R group is different. Hopefully you see the change there, right? Much different R group. The third one, let's see here, is also different. The third one is different in the altered sequence. The fourth one is the same, right? It's the same identical R group. And the fifth one is also the same. It's just a hydrogen. So we should keep that in mind as we go into looking at the questions, that it's these two middle amino acids, 2 and 3, that have been changed. Okay, now we can look at some of the questions here. Now, I mentioned in class that our first tip for answering free response questions is to identify those task verbs. And they're already bolded in here just to help you see them clearly. But this really helps us to make sure that we're not doing more work than we need to, and to make sure that we're actually answering the question that's being asked. So the first question here is identify the process used to form the covalent peptide bonds that join amino acids into a polypeptide. We know this, right? This is the process that builds polymers. We call this dehydration synthesis. And the question is just asking us to identify it, so we don't need to explain it. We just need to identify it. Question B. The change in the amino acid sequence illustrated in figure 1 caused a change in the shape of receptor X. Based on the R groups of the original and substitute amino acids, explain why receptor X changed shape. Okay, so for that, we're going to have to take a look back at the R groups that were substituted. Okay, so I copied in those amino acids again right here. So remember that it was amino acid 2 and amino acid 3 that had changed. Amino acid 2 and 3. So let's look at the properties of the R groups of these two amino acids. So for the normal, notice that this R group has a positive charge. It's got a positive charge right there on the NH3. And so right away, that should be telling us this is a charged R group, and anything with a charge can interact with water. So this is likely going to be a hydrophilic amino acid that has a positive charge to it. Now, when it was altered, let's see if they have similar properties. So down here, we notice that there's no longer an NH3, and there's no longer a positive charge. In fact, it's just CH and CC bonds found in the R group. And we know that those are non-polar, right? So now this is a hydrophobic R group, whereas this one was charged and hydrophilic. And I'll put uncharged down here as well. Okay, so that's a significant change in the property of the R group, and that's going to affect how this protein folds. But let's look at the third one. So here, this also has a charge of a negative charge right there. This is also a carbon-oxygen bond, which is a polar bond, and so we're also going to see at least partial negative and partial positive charges. So this is a charged hydrophilic R group as well. But we notice that it changed again to a CH3, which is a hydrophobic and uncharged R group as well. So in both situations, we've gone from hydrophilic to hydrophobic. And we know that hydrophobic R groups, they're going to want to avoid water. And so they actually kind of move towards the inside, away from the outer edge of the protein where water is found. And so this is going to really affect how this protein folds, especially in the tertiary structure, because of these changes. Okay, so just to summarize what I just said, the shape of the receptor X protein will change because the substituted R groups have much different properties. In the normal amino acid sequence, both R groups were hydrophilic and charged, but they were changed to uncharged hydrophobic R groups. This will cause the protein to fold differently. And that's our explanation. Okay, now moving on to the next question here. It says, using the template below represents the 4 levels of protein structure. Place an X on each level that is expected to be altered as a result of the amino acid substitutions in figure 1. All we need to do here is not explain, but just place an X. So first of all, we know that the primary structure was definitely changed because the primary structure is just the amino acids connected by peptide bonds. So we had 2 amino acids substituted. So the primary structure was definitely changed. Now, looking at the other ones, we have to think about what part of the amino acids are involved in making the secondary, tertiary, and quaternary. So the secondary, if you remember, involves only the carboxyl and the amine groups. We didn't change any of that. And based on what we read as well, didn't sound like there were any beta pleated sheets or alpha helices to begin with. And so because we didn't really change any of that, when we substitute the R groups, the secondary structure is not going to be changed. But the tertiary structure where R groups interact, that'll definitely be changed. Because now we've got hydrophobic R groups instead of hydrophilic R groups. That's going to affect the tertiary structure. The quaternary structure, again, is multiple polypeptides interacting together, but from the context, it told us that there's only one. There's a single polypeptide that makes up this protein. So the quaternary structure doesn't exist for this protein. So our answer is primary and tertiary. Okay, so the last part here, it says explain how the amino acid substitution is most likely to affect the function of receptor X. And what was the function? Well, in the reading, it said that it binds a molecule that's found outside the cell. And when it binds that molecule, it causes the cell to respond in a particular way. And so for this, the structure, since the structure is changed due to the new R groups, right, and we explain that in part B, the molecule that binds to receptor X likely won't be able to bind anymore because of the different shape of the protein, right? And that's going to ultimately affect its function, right? So we came full circle. Likely this receptor won't function because it's folded differently as a result of these substituted R groups. So that was the first question. I hope that was helpful. Okay, so let's go over the second free response question, which is a little bit trickier. So the second one, let's start with the context. It says white blood cells cause B cells produce proteins that can be used for the treatment of certain illnesses. However, these B cells do not live for very long on their own. To keep the B cells growing for a long time in laboratories, scientists fuse the B cells with cancer cells. We call that fused B cancer cells that do grow for a very long time. The particular cancer cells used for the fusion are treated with chemicals that make them unable to produce the nitrogenous bases adenine and guanine. But the B cells with which they are fused do produce these nitrogenous bases. The scientists grow the large fused B cancer cells in a growth medium that contains necessary nutrients for the cells and includes a source of carbon. Now, right off the bat, we should see the purpose. So the purpose is that something to do with these B cells producing proteins that can be used to treat illnesses. But they don't live very long. And so to help them live longer so they can produce more of those proteins, they fuse them with these B cancer cells, or with cancer cells to make fused B cancer cells. Now, when they do this, they mentioned also that there is this complementary nature of these cells because the cancer cells, they are not able to make adenine and guanine, which are the purines in DNA. But when they're fused with B cells that can, all of a sudden they have what they need, and together they can create a fused cell that not only allows the cancer cell to live, but also allows the B cell to live longer. Okay, so now that we have a little bit of an idea of the context, let's look at the questions. Okay, so the first question here is to describe the role of carbon in biological systems. And we're familiar with this because carbon, remember, has a valence of 4, which gives it lots and lots of flexibility in building macromolecules. And so this question is mainly just asking that, right? That carbon is used to build many biological molecules, including, and you can list out the 4, including lipids, proteins, nucleic acids, and carbohydrates. Okay? And that's it, right? That carbon it's central because it's needed to build biological molecules. Okay, the second question says, The membranes of both the B cells and the cancer cells are largely composed of phospholipids. Explain how when the membranes are fused, which means combined, the polar parts of the phospholipids from one cell will interact with the phospholipids from the other cell, and how the nonpolar part of the phospholipids from one cell will interact with the phospholipids of the other cell. So in your reading, and also in Mr. Anderson's video on biological molecules, he talked about phospholipids, and this is just a simplified model of one. Phospholipids, they have what are called a head and a tail. The tail is mainly carbon-hydrogen bonds and carbon-carbon bonds, and so it is mainly nonpolar, which means it's hydrophobic. But the head has a negative charge due to a phosphate group, kind of like we see in DNA. And so because of that, it is hydrophilic, because it can interact with water. And so when you put these two cells together, their membranes are mainly made of these molecules. And so we can also think about how in proteins, hydrophobic R groups tend to hang out with other hydrophobic R groups. And so that's kind of the same thing that happens with these phospholipids is, and this is towards the question, the hydrophobic portions of the phospholipids will arrange next to other hydrophobic regions. And the same will happen with the hydrophilic heads. And so what will happen is you actually get the membranes fusing together into something that looks like this, where the hydrophilic heads are arranging and sticking together, and the hydrophobic tails are sticking together. And you start getting this barrier. And you'll also get, and we'll talk about this next unit, what's called a bilayer. And you notice that all the hydrophobic portions are sticking together and the hydrophilic portions are sticking together. So that's what we're looking for in that question. Okay, moving right along. Question C, make a claim about the most immediate effect of the fused